Dear friends, thank you for joining us today morning for Department of Medicine academic morning breakfast session. Once again, we are thankful to Dr. Mahesh Moorthy, Professor of Virology from CMC Vellore. A couple of weeks ago, he took a session for us on the PCR testing for COVID. Today, he's going to talk about the immunity to COVID. And we are very thankful, Mahesh, that you accepted our invitation. And we are very grateful. We had thoroughly enjoyed your first session. And we are looking forward to one more academic feast today. So thank you very much. For the people listening, I would urge you to stay till the end and write down your questions. If you remember from last time, Mahesh patiently sat back and answered so many questions which people had. So please put it in the chat box and we will make sure that all your all your questions are covered. So it's the middle of the pandemic and a right time to learn about the immunity to COVID. So thank you again, Mahesh. Thank you to Mohan Jambugalam for organizing and the telemedicine team, all the members there for arranging this. So without much ado, over to you, Mahesh. Thank you.
Many thanks to Dr. Tambu. It's a great opportunity to present this talk on immunity to COVID. Now, can I have the presentation? Thank you. My last talk was about more to do with detection, though it said lab diagnosis of viral infection. It was more to do with detection of the virus per se and more to do with PCR and direct detection methods. In this talk, I'm going to try to focus on more to do with immunology and specifically on serology, because that's what is the burning question now, and what are the methods, the questions about what methods we're going to use and how we're going to deploy them and all of that. I won't touch on too much about cellular immunology because that's an evolving field, in fact, very rapidly, and I think we will probably, if there is an option, maybe another session on that as well, because it's quite a very, very interesting field that's changing. So you may all have, in different roles, you would have been part of this pandemic, either as from the clinical departments or from the laboratories or as support staff, and we know that it has pretty much overwhelmed us in many aspects, and we are either a COVID hospital where we do COVID related work or we do non-COVID related work. We've kind of split up into two kind of parallel streams. So I just want to quickly take you through what data we have from our initial testing services that we've offered. It's not really data, it's just things put together. It's just for us to get a perspective of how bad this is. So if you look at our peak, there was nothing much in the early weeks of this pandemic, and this is from early April all the way through to August 15th. So there wasn't much in the early, if you can see my pointer, there wasn't much, very sporadic detections here and there, but it was towards the end of June that it's really hit us and things started picking up, and you can see the detections are fairly high across all age groups. The key factor here to note is that the very young, or up to 20 years of age, there's fairly few detections. I don't know if that is an artifact or whether there were lesser samples, but it's pretty evident that there are fewer detections, and that's something that we should explore when we're looking at clinical data as well, in conjunction with this data. So the number of symptomatic SARS positives are at least 5 times the asymptomatic, and it's quite evident in this heat map, and we've done close to 17,000, probably running on 18,000 samples now. So it doesn't seem to be slowing down, and we are consistently maintaining the same level. So the next thing that will come up on everybody's mind is, what is the next step? What do we do? What next? Because you may all be aware a lot of staff have been infected, and we're trying to look at how do we maintain our services. So just to give you a quick recap of what laboratory diagnosis, essentially the key methods you can use to diagnose SARS-CoV-2 infection is either direct methods, which I won't touch on, it was part of my previous talk. More to do today with indirect detection, where I'll tell you mainly about antibody detection and briefly about cellular responses. So indirect methods are methods to detect an immune response to the virus, and so far this is a pandemic, and a lot of things you would have heard the word EUA very often, emergency use authorization. So there are so many kits and so many different kinds of devices that are available, and all of them, and it's a daunting task to choose the best one. So there's the key strategies have been to look at the spike or receptor binding domain and the N, and with either an ELISA format or a chemiluminescent immunoassay format, or sometimes rapid devices, though that's not really recommended, but it has use in some settings. So you must be quite familiar with this, the top figure up there. You know that there are asymptomatics, there are pre-symptomatics, the mild infections, and there are severe, and we face a lot of severe as well, and there's been a fair amount of mortality. If you look at the kinetics, not so much about the direct, but if you look at the antibody here in this figure, find that this is only a schematic, it's just a cartoon, but the real world data pretty much mirrors what is seen here. You find that IgM and IgG are pretty much co-occurring, but IgG, immunoglobulin G, tends to persist for longer. And people have also been looking at IgA because it's a mucosal antibody. Okay? And you must remember that though you can have RNA positivity, which happens very likely during the early stage of the illness, or sometimes even in the asymptomatic or pre-symptomatic stage, and very often in the symptomatic stage, but you find that you will get antibody tests negative early on in the disease. Say that would probably be up till 10 days of illness from symptom onset. And all of the devices that we know about, all the kits we know about, have always had this stratification of, accuracy indices less than 10 days of illness and greater than 10 days and so on, and beyond 21 days. So it's obvious that the accuracy depends on what time or when in the illness you're actually sampling and testing for antibodies. Okay, so the first method is an ELISA, an enzyme-linked immunosorbent assay. Now, you must be familiar with what an ELISA is, because it's used very, very commonly. It's used for a wide variety of infectious diseases. And this is a pandemic, it's a novel virus, so everything was developed from scratch. So we are at the stage we are in, we are pretty much in the stage of, the first generation HIV ELISAs, where, initially there was a lot of problems with the assay, and with time, with over many, many iterations, we find that now the HIV ELISA is really good. But now we are at a stage where we only have the preliminary first generation of assays that have come out, and mainly the key focus has been receptor binding domain of the spike and the spike, either the whole spike or the S1 and S2 and the nucleoprotein. The reason for this choice is, if you look at this table on the right, you'll find that this table kind of is an indication of how similar each of these proteins are across all of the strains that belong to the human strains in the Coronaviridae family. You find that very close to SARS-CoV, the original SARS virus, okay, across the board for all the proteins, but S1 seems to be a little bit better. So the choice is, if you want to differentiate between SARS-CoV and SARS-CoV2, an S-based assay is probably a better bet than an N-based assay, because N-based assay is quite close. And the reason that S1 is chosen so often is because it's quite divergent from the other, you can see from MERS and also the other seasonal coronaviruses. So these are the key assays that are being used. And so when you look at an antibody detection assay, primarily it's an indirect kind of method where you coat the antigen onto these microtiter wells, which are very small wells. You coat the antigen on the sides of the wells, and you add your serum, which will have antibody to it. If the serum has antibody, it will bind to the antigen that's present on the well, and you can, based on what antibody you want to detect, whether be it IgM, IgG or IgA. You can vary the intermediate reagent that is used, which is a an enzyme which is either horseradish peroxidase or any other enzyme which produces a colorimetric reaction. So if you want to choose an IgM assay, you look at anti-IgM tagged with an enzyme, or if it's IgG, you look at an anti-IgG, and so on. So these ELISAs are mainly initially developed as in-house ELISAs, and the commercial companies have joined the bandwagon, and there are lots of assays. There's close to 300 or 400 assays that have been developed. Now this is a big problem, because you can have an in-house assay which may or may not perform as well, but then you have a commercial assay and the manufacturer claims that it's a fantastic assay and it's doing very well. So how do you choose? So which is why at any point before you deploy an assay, before you test it, before you want to offer it either as a service or as research or anything else, you would want to test it out and see if it is as good as the manufacturer claims. Okay. Between the S and the N, the S seems to have more sensitivity, but there is some utility in, be it the IgM or the IgG, but there is some utility in having the N as well. And there have been some papers now that have described it as a kind of divergence where an immune response directed more towards N is associated with a worser outcome, and an immune response directed towards an S kind of where antibodies are directed towards S, so they form the majority, kind of a ratio of S and N, lead to a better outcome. So all of this is preliminary, and most of these are preprints, but we will see once more data comes out, and possibly we might even explore doing this kind of work in the institution. Okay, so as I told you earlier about HIV ELISAs, where you found that there were initially virus lysates, or first generation ELISA. Modifications of them were those that refined the reactivity in such a way that only a specific portion of the proteins of HIV were used. Likewise, for SARS-CoV2, specific peptides of the S and the N were used, and this was tried out. And this system, where they used 3 different peptides from S and N, seemed to differentiate mild from severe. So that if you use a peptide ELISA, you find that there's less noise and there's less cross-reactivity. So you're actually able to use it, and it has some utility in determining whether differentiating between severe and mild disease. But you find that in severe you tend to have more response, in a milder disease you may not have that response. The other assay that's used fairly frequently is the chemiluminescent assay. Now the chemiluminescent assay is made for throughput, made for ease of use, so all the big corporations have joined in, and they want to offer these ELISAs for everybody to test. It's great because they have a very good throughput. The results are available really quickly, but there is a problem. They are not as reliable, they are not as good, and again, going back to, as with any ELISA, even a CLIA needs to be evaluated to see which one performs the best, because when you have low sensitivity and or less than perfect specificity, you have to deal with either false negatives or false positives. Okay, the chemiluminescent assay essentially is based on a light readout, and that contributes to its higher sensitivity. It's claimed that it is more sensitive, and we find that that is truly the case, and add to the fact that it's automated, so the hands-on time required is by the technician is lesser, so it's a great system to have. We from CMC Virology have been exploring a few CLIA assays because we already have the setup, and we will try and finalize one of these, either the Siemens, Roche, Abbott or the Liaison, and all of these are being evaluated just to see how well they perform. And okay, the next real assay of importance, which is considered a gold standard when it comes to antibody detection, is the virus neutralization assay. The virus neutralization assay essentially determines the ability of antibody to prevent an infection. It prevents virus entry by blocking the surface glycoprotein that binds to the host receptor, and therefore there is no chance of entry into the cell. Okay, so in a virus neutralization assay, it's essentially an assay to look for antibodies in serum. So serial dilutions of serum are used, which when mixed with a standardized amount of virus tend to, if there is specific antibody, it will inhibit the entry of the virus into the cell, and the end point is the development or the inhibition of viral CPE. The development of CPE is because there is no antibody, and therefore the virus can enter into the host cell. If there is antibody, it will, and it's neutralizing in nature. So you must remember that neutralizing means that it is able to inhibit the infectivity, but you can also have non-neutralizing and just binding antibodies, which are directed towards the regions that are not involved in the interaction with the host receptor. So for example, you have other regions of the spike, or you have regions of the N which may not be neutralizing because they do not interact with, they do not come in the way or have no hindrance to the interaction of the spike with the receptor on the host cell, which is in this case ACE2. The problem with this is that it's quite cumbersome. It is difficult to do. There is the need for a BSL-3 facility because it is a high priority agent, so it needs to be done in specific facilities and adequate training, because you're dealing with live virus which you're going to use again and again. An alternative which gives a good readout. So I said that earlier that the readout is viral CPE for the classical virus neutralization test, but if you want to use a method where you can count the plaque forming units, what is done is in the same system you overlay the cell culture cells, the host cells, with agarose or some low melt agarose, and what that does is it ends up preventing the virus from spreading all over the monolayer. And therefore when you stain it, and this here, the blue stain here, the regions that are unstained represent plaques. A plaque is akin to a bacterial colony where each individual virion will go on to infect neighboring cells and grow larger. Okay? So the plaque reduction is the percentage or the fraction of reduction of the number of plaques that occur when you add a certain amount of antibody, and there's a 50% cutoff. So that is a better readout. So because of better readability, that's also followed. Again, even that has the problem of lower throughput and also needs to be in a BSL-3. The focus reduction neutralization test essentially is a similar method, but the readout here is a fluorescent based, where you have antibodies specific to the virus. So after doing the whole infection, we stain with antibodies which are tagged to fluorescent dyes, and therefore the readout is fluorescence. And once you have fluorescence, you obviously have better sensitivity of the assay, and you're able to pick up even weak signal. Virus neutralization, as I said, is a gold standard, and it's more relevant in a vaccine evaluation setup or for development of therapeutic agents, because it's not feasible to do things on a larger scale. Variations of the neutralization assay, and the big drawback of the neutralization is that you need to have a BSL-3 facility, which are expensive to set up, expensive to maintain. So therefore you need something that you can do at a BSL-2 level, which is basically what all of the lab work that we do when we do PCR. So that is great if you have that kind of a setup. So pseudovirion based neutralization assays are another option. A pseudovirion is essentially a lentivirus. A lentivirus is basically belongs to the family retrovirus that HIV belongs to, and they have the property where when they can acquire different proteins of heterologous, they can acquire genes of heterologous origin, and they can still bud out and form virions. So this virion is called a pseudovirion because it's not really the all of the other parts of the virus are the lentivirus or HIV or VSV as the case may be, but the only thing that is different is the glycoprotein, and in this case it is a spike glycoprotein. So you find here that if you look at the figure, there are different proteins that code for all the components of the lentivirus, and you have a plasmid that encodes the spike of SARS-CoV2 and a lentiviral backbone with a fluorescent signal. So when you mix them all together in a mammalian cell culture, you find that they tend to when all the plasmids together infect one cell, it's basically a transfection experiment, and once that is done, you find that there is a production of virions, which are actually pseudovirions, and these pseudovirions can be assayed for. How the pseudovirion neutralization assay works is that once you have developed your pseudovirion assay, you also need a cell line that expresses ACE2. ACE2 is the receptor for spike. So when there's a cell line that expresses ACE2, right here, the cell line expresses receptor for the spike, and when you infect this cell line with the lentivirus, the virus will enter the cell, and once it enters the cell, it has certain promoters which produce fluorescence. Now when you react this lentivirus with antibody, which is neutralizing in nature, meaning it binds to the spike, it will prevent this entry, prevent this interaction of the spike with the antibody, with the receptor, and therefore prevent the entry. So there you have it. It neutralizes the infectivity, though it's not infectivity per se of the SARS-CoV, but it's a demonstration of the fact that the antibodies bind to the receptor binding domain of the spike protein. There are variations of this. There's one which is the high throughput assay, where you use a modified SARS-CoV2 which has one of the genes replaced with a fluorescent reporter, but that's a high throughput assay where it needs BSL-3. So that is more useful, one, for antibody, but you can also use it for drug screening, because you need to look at large number of compounds. Other assays that use the same neutralization principle, there's an assay called the surrogate virus neutralization test. Now, the key principle of the neutralization is that it needs two components. One is the spike protein and one is the receptor. So instead of having a cellular system, you have a system which is on an ELISA plate or on an ELISA well. So you have the ACE, the receptor, bound to the ELISA plate, and you add conjugated receptor binding domain. So you find that if there is no antibody in a serum sample, you will get because the conjugate will bind to the ACE2 and produce a colored reaction. So if there is no antibody, this colored reaction will proceed and you will have a very bright signal. But if you have antibody that prevents the binding to ACE2, it basically neutralizes it. It shows that there is neutralizing antibody present, and it's seen that it correlates quite well with PRNT, and this is even better than the other pseudovirion assay, because it's a simple ELISA format. It can be done really quickly. Another one is a nanoluciferase biosensor, where basically the RBD and ACE are tagged to a luciferase receptor subunits, and when they come close together, they produce light. When they don't interact due to the presence of antibody or due to any inhibitory compound, So I'm kind of confusing it a little bit by saying inhibitory compound, but essentially it's the same principle. Okay, so you want to prevent the binding of the spike to the ACE, and therefore that is an entry prevention drug or a candidate. So a neutralizing antibody acts in a similar manner, and it prevents the entry or prevents the binding of the protein to the receptor, and there will be a reduction in the light, or there will be no light produced. Okay, I'll move on to immunochromatographic tests. So these are basically rapid bedside point of care tests where you're trying to look for specific antibodies. There have been devices for antigens as well, but that's that I talked about earlier in my previous talk. But these devices are used to detect both the IgM and IgG, basically either the blood or saliva or any respiratory fluid is dropped onto the sample pad, and it reacts with nanoparticles which are antigen tagged. So if there is antibody, it will bind to the nanoparticles, and these nanoparticles, these antibodies are captured by an anti-IgM, which is here, or an anti-IgG. And in the case of the control, which is the last line, just to say that the fluid has traversed the entire length of this particular device. The control is usually at the last. So you find a anti-species antibody which is bound, and therefore it's an test adequacy indicator. The final reaction, because it's a nanoparticle, it's got gold nanoparticles, it's usually a visual readout. You can actually see the color. So you may have heard about this when we used to do, many years ago, used to do dengue devices. We used to say weak positive, very weak positive, very, very weak positive, because that's what we see. So there's a problem in interpretation in all of this. These devices as such have a poor sensitivity, 30 to 80%, depending on the time frame, but they have some utility in specific scenarios, but cannot and should not be used for diagnosis of SARS-CoV2 infection, either in an admitted patient or in the context of seroprevalence studies, because again, poor sensitivity, meaning that you can have a lot of false negatives, and also the specificity can be quite poor. So these assays have been, there's been close to there's been cases everywhere, and we in India are struck by a very large number, but we haven't done this level of antibody detection that is expected, that will help us to interpret what actually is happening. But there have been a lot of studies in numerous countries, and they've tried to look using these particular assays, either the SRBD or the ELISA, have tried to correlate and see if there is some correlation between disease, severe disease, and the level of antibody. Okay. It was found that severe disease produces higher titers of antibodies, and whether it be by ELISA, which is checking for the RBD, or SNN, or neutralization assay, you found that there's always higher titers in severe disease. So in severe disease, antibody detection may actually be useful in trying to diagnose disease. It was found that 20% of the RBD ELISA, okay, that is out of 100, 80 of the RBD ELISA positives actually neutralized virus, but the remaining 20% didn't. Meaning that even though you're saying you're using a surrogate of neutralization, or if you want to say that the RBD is the closest you can get, antibodies to the RBD are probably the closest you will get to an actual neutralization assay, you still find that there's 20% loss there. The N antibody is little worse off, and it's only 25% of the that is 75% correlated with neutralizing antibodies. Okay, so there you find that there is a problem with, yes, you can diagnose, but then when you're looking at kinetics and looking at it in the longer term, there will be a problem, and especially when you use it in convalescence and you use it for seroprevalence, you'll find that there's different kinetics, and therefore there's a problem in what assay you use and how it performs. The neutralizing S and RBD are fairly, they stay for longer, they're more gradual in their decline, but the N seems to fall off much lower. But even among the neutralizing antibody, you find that even if you start off with high titers, there's fairly quick waning of neutralizing antibody, and at this particular study, they found that at about 95 or 100 days, only 16% of the samples which were originally high titer, and in this case, greater than 1000, the dilution factor for neutralizing antibody, only 16% remained high, meaning that there is waning, and if you start off low, you will have much faster waning. They've also explored saliva as an option, and saliva has been used for detection of antibodies, either using an IgA format or using a transudate, which is basically try to see the transudate of the serum and look for IgG. In saliva, we find that the antibody is similar between serum and saliva, okay, and the kinetics are similar between when you look at IgG, and it rises close to the second to third week of infection. Now, serum IgM and IgA decline, but you find that the IgA in the mucosal compartment tends to stay a bit longer, so we may actually have a surrogate of systemic immunity in the mucosa. I also found that IgA levels in the acutely ill patients were higher, so if you have an assay where you have PCR negative and you find that IgG is also maybe negative or indeterminate, perhaps looking at IgA would be an option, where if you look for IgA, you may actually be able to diagnose presence of antibody. Okay. So the serological recommendations. So as of now, what IDSA recommends is that it only be used in epidemiological studies and in situations where there's a high clinical index of suspicion and repeated molecular diagnostic tests coming negative, but the clinical picture based on very stringent criteria is very suggestive. They specifically categorically mention that it should not be used to determine immunity or risk of reinfection for numerous reasons, because you don't know, and there's actually in fact only one study that has actually documented this, and that's come out as a preprint a couple of days ago, which says that neutralizing antibody is actually protective, but you have to look for neutralizing antibodies. So serological tests, meaning the non-neutralizing ELISA based assays, should not be used for determining immunity. It also should not be used to inform decisions of discontinuing physical distancing and lessen the use of PPE. The recommendations from ICMR are on similar lines, where serology is currently recommended for use in seroprevalence studies and also among healthcare workers, where the high-risk group, to check for positivity. It is not recommended for routine diagnostics, and I think currently it's in CMC is only being done on a research basis, where we're trying to refine diagnostic algorithms to say which is the best choice of tests or combination of tests that can determine the infection with SARS-CoV, be it PCR be it PCR and or antibody detection. Okay, so I showed this slide as well last time, and would it be possible to kind of zoom in, because I'm not sure if it's very clear. It's not clear to me. Okay, if you see the red, you'll find that this is a graph of sensitivity versus specificity, and these are the assays that you actually want to use. Okay, but all of these seem to be working very well when it's the late stage of the disease. And this is antibody detection overall, meaning it is all assay formats, all types of antibody, okay, and all durations of illness. Okay, so you find even among the late there are assays that perform quite badly. So it's something that we should tread very carefully when you want to make a choice of device. We have 4 options currently, and we probably will go forward with 1 or maybe 2 of them. But then again, it depends on what is the utility of it. So, as I showed in the previous graph, just to summarize the serology, it has higher sensitivity when done later in the disease. Specificity is high, but most of the evaluations of specificity have been done on pre-pandemic samples, where you say, you test samples which are before the pandemic, but they haven't been tested in the context of the pandemic, where you try and look for specificity of those who don't have COVID. So that's a bit of a skew there, but going forwards, when more numbers are tested, you find that more data will come out, which will give a more accurate estimate of accuracy. They have a role in RT-PCR negative cases, and seroprevalence used with caution, because depending on what assay you use, false negatives and false positives will occur. So in mild disease, as I told you earlier, that when you look at ELISA RBD, there's a discordance with neutralizing antibody. But you find that in neutralizing antibody itself, those that are proven PCR positive, you'll find that they are negative. So only 90% of them with mild disease will actually produce antibody. Okay, going forwards, key factors like the correlates of protection. What is it that determines whether you're protected? I can give you the example of influenza, where you have the HA, and you don't say any HA antibody present is protection. You have specific set criteria, which is either 1:40 or 1:80, as the case may be. So it's important to find out what is that correlate of protection. And even for flu, there is a problem where we don't really know, because serum antibody is one thing, which is what is measured, and the infection or the risk of infection is actually at the mucosal surface. So is a mucosal antibody a correlate of protection? That's something that should be explored. And the way it's going, I think IgA would be a good candidate for routine diagnostics, as well as try and see if there is some correlation with protection. Correlates of progression are not clear. There is some indication that severe disease have higher levels of neutralizing antibodies. So that itself, or a titer in itself, or a semi-quantitative value may not be sufficient, and we would likely need to have more clear-cut criteria to say what are the correlates that say that you will progress to a severe disease, or you will end up with a severe infection. Now, there's only one study, which is a preprint, which shows protection from reinfection, where they tested people going on a boat who went out into the sea for about 30 to 35 days. They tested them before they left, and for PCR as well as antibody. And when they came back, there were multiple infections, multiple samples were collected, and all of them were sequenced. And the 3 people who were neutralizing antibody positive prior to the journey did not have any evidence of infection, did not have any PCR positivity, and their antibody titers continued to stay high. So this is probably the only evidence that we have that neutralizing antibodies when looked for, and again, that's a quantitative or semi-quantitative estimate that needs to be determined. will probably give a protection from reinfection. Serology has utility in familial transmission studies where sometimes PCR can be negative, but when you sample them in a community setting, in a familial transmission setting, you find that the serological methods tend to perform better. Okay, I'll end with just a quick overview of the T cell response. So there's nothing new about what's happening with SARS-CoV2 and how it affects the immune system. What's different is the scale to which it's happening. Now you expect that in any viral infection there will be activation of all arms of the immune system. You will have an innate immune activation as well as the cellular arms will be activated. So the key parameter here is the CD4 cells. Likewise, you also have CD8 and natural killer cells that contribute to, in some way, the pathogenesis and to the severity of the disease. We found that it was as expected. You found that there was a activation of CD4 cells which reacted to S peptides, that is peptides from the spike protein, and they were of a memory phenotype. There was also reactivity to N peptides and to patients who recovered from SARS, the original SARS. The key feature here is that 2 or 3 of these studies have shown that they have shown reactivity of unexposed individuals, people who have not had SARS-CoV2 either by PCR or who are negative for antibody, actually have a cellular response, either CD4 cells which are reactive to certain peptides which are shared. Okay, it's most likely due to shared peptides between SARS-CoV2 and the seasonal peptides. More exciting work will probably be coming out soon, but for now I will stop with that and I'm happy to take any questions. Thank you.
To request all the participants to drop your comments and questions in the chat box so that Sir can answer. Thank you.
Okay, there's the first question. What type of test is being used in Delhi to say that 29% have antibodies against the virus? Are they neutralizing antibody? So I don't know what is being used because it has not been put out. Maybe there is a preprint, I'm not sure. I have not looked, but it's very likely that it is probably an ELISA that's being used. So then there's also the problems with the ELISA. If there was neutralizing antibody, I think they did a fairly large number, so it's unlikely it was neutralizing antibody because those take a lot of time and it's quite cumbersome to do. It's very likely it was an ELISA. No more questions. Great. What is the role of the new saliva test? So the new saliva test is, so we are exploring that, and the saliva direct is an assay that's being used for PCR detection, which is for detection of the virus, and that has the advantage that it doesn't need this extended extraction step. So that's from the saliva is being used from the direct detection point of view. We have not started off with can you just go up? What is the second question? We have not started using the saliva assay, or we don't plan to look for saliva antibodies, but probably we can look at it from a research angle and try and see if they match serum antibodies. So I'm guessing our priority now is to start testing serum, and that too in healthcare workers, because that's the priority and that's what's allowed. But in a research setting, you can probably look at things like saliva. I think I answered Simon's question as well. It may be inappropriate to say that if 50% of Delhi's population have antibodies, they will have herd immunity. It was in today's Hindu. Well, it's the percentage that's reported is a function of what assay is used, and it may not be very far off the truth. Okay, the rates of false positivity are a little low. It's not really that much of a concern. If anything, it's probably in this range of 50%, but it's we don't know the details of what was done, so I can't comment on what exactly the performance of that device is, of that test is. Are there any large sero surveys in India in assessing the community prevalence at a national level? So I think ICMR has undertaken a lot of these surveys in India, and that data is not out in the public domain, but I think they are analyzing it, and these come out as press briefings and things like that. But there are large studies that I think Vellore is also a part of from DBT, which will look at trying to assess sero incidence, trying to see temporal sampling and try and see what the burden of SARS-CoV2 antibody is. Right. Not related to diagnostics, but the concept of RNA DNA vaccines and it continues to target proteins can hypothetically induce desensitization. What is its impact on vaccination and serology measures? Okay, so there's a lot of schools of thought which are talking about more not desensitization, but there is a fear of antibody dependent enhancement. It's still not yet been seen with natural infection, but when you do have vaccines coming in, now RNA DNA vaccines are not so much prone to this kind of side effect or this kind of phenomenon, but you find that when you use live virus which was then inactivated and then that was used for vaccination, that can have a problem, and we know this with the RSV vaccine trials where enhanced respiratory disease happened because of vaccination. But we don't know. I mean, it's still out there. We don't know what's going to happen. Okay? And I don't know how much of vaccination is actually going to happen for it to affect on such a large scale. Will the new mutations of the virus interfere with these assays? Okay, so there are some new key fixed mutations that have been described in lineages all over the world. We don't know if those lineages have come to India, but it's very likely they will, and eventually that will also happen. The thing about it interfering with these assays, it's likely that if this mutation is exactly in the receptor binding site, yes, there will be some interference, and what assay we are planning, which is based on an older virus, may not actually work. And people are well aware of this, and there is in fact work that's going on to say that there are specific sites in the RBD which are prone to mutation, and therefore this should be taken into account, and there will be probably upgradations of assays as we go on. Is the salivary IgA any value in protection? We don't have the answer to that. We can't say protection, but we can say present or not present, and it's possible that we could check it among healthcare workers. The thing is, we don't know about protection until there is a specific study that's been done. So once that is done, we can say that it does protect or not. And I think many studies will come out soon. It's just a matter of time. What is the prevalence of D614G in India? I don't have that answer because there is that data is still coming out. There's recently been a 1,000 Genomes Consortium which have sequenced 1,000 genomes, which is coordinated by DBT, but I don't have access to the data and I have not seen the preprint. So we don't know that yet, but I'm sure it's there. What will be the final role of antibody test? Yes, I think currently if we have to look, if we have to say, it's probably seroepidemiology and healthcare workers. But eventually, I think going forwards, and that stand is, it's only a matter of time when assays get better, and we are allowed to use these assays for routine clinical diagnosis. And that's, I don't think that's very far off. It might happen sooner than we think. What is the role of T cell response in protective immunity? Will it provide more lasting protection than neutralizing antibodies? Okay. So what we only have now is that we are trying to say there's a difference from an immunologist's point of view, and there's a perspective of a clinician or a person who's in the community. An immunologist look at reactivity, where it reacts to a particular peptide, reacts to a particular protein, and therefore you're able to say that those T cells are present. Whether that translates to protective immunity has to be done, one, in animal studies and also in long-term follow-up cohorts. You can't say T cell response and protective immunity is a given immediately. Okay? But that said, there is evidence that the T cell response from seasonal coronavirus seems to be reacting to or is present and reacts to peptides of SARS-CoV2 in spite of the patients not being exposed. So if anything, and what we know from flu and other viruses, is that T cell response will at least lessen the severity of disease. So if we're finding this and it is more widespread than we think, then it may actually dampen the effect of the epidemic in terms of severity. Neutralizing antibodies, it seems to be that they don't seem to last very long, but it's still early days, and we might have better studies that will tell us if that will change. Are we seeing reinfection? If not, can we say that antibodies are protective? To be able to say reinfection, you need to be able to see the person the second time and then say, okay, they got infected the first time and then they were positive and then they got an infection again, another episode. So you need to have the time to actually look at the second patient. And I know nobody has the bandwidth to do that kind of work. But reinfection is a possibility. We know this from seasonal coronaviruses. We don't know if it's a certainty, but we know that it's possible because seasonal coronaviruses seem to have antibodies that are not very long lasting, and you can have reinfections that, I mean, these were papers from the 70s, from the 60s and the 70s, so they may not actually be very, very accurate in the real world today, but we do find waning of response and reinfection with seasonal, so it's likely it would happen, but we don't know. We'll just have to wait and see. Antibodies being protective, neutralizing, if checked for, there is evidence that it is protective. So I think we end that. That was the last question.
Mahesh, that was absolutely fascinating. I really loved it. And I'm sure the audience also was enthralled by your presentation. Terrific, terrific. Thank you so much. Thank you for staying back and answering all our silly questions. I'm sure they must have sounded very basic to you, but thank you for being patient with us.
Thank you for the opportunity, and I hope I can probably come back at some other point, maybe not in the near future, but at some point, and talk about the cellular aspects of it.
Yes, we'd love to hear the T-cell aspect later on. So thank you so much, Mahesh. And to the audience, thank you. Thank you for staying back and listening to the question answer session. We have an exciting series of immunology talks by Dr. Danda and team from the next week onwards. So those of you who are interested in immunology from a physician's perspective or from a medical perspective, please join us for the next couple of weeks. We're going to be having immunology talks led by Dr. Danda and his team on different aspects of immunology, because it seems that's an interesting topic to learn now. So with that, once again, thank you to the telemedicine team. Thank you to Mohan. And thank you for all of you for joining us. Again, what we do is for God's glory, and we are grateful for this opportunity. So thank you again, Mahesh. Deeply honored that you
Thank
would
you.
accept our invitation. Thank you, Mahesh. All right.
Thank you.
Bye, everyone.